Welcome to Cap 5. This demonstration will take you through the main features of our program. The first demonstration dataset we will use is of river invertebrates collected in chalk streams in southern England. This dataset is supplied with CAP. All of the demonstration datasets are stored within a folder called Demo Data. The data are stored as a comma-delimited text file. You can open one of our standard datasets in the spreadsheet of your choice to see how the data are organized. CAP will check for problems when loading a dataset. For example, if one of your variables holds no data, CAP will exclude it from the analysis. Click on the Raw Data tab to see the data that you've loaded. The data are arranged with the columns as samples and the rows as variables. For the stream dataset which we have just opened, the variables are the abundances of the families of various insects and other invertebrates. They might also be the abundance of species, or perhaps the concentration of various chemicals. Above the grid are various controls that can be used to edit or rearrange your data. Click on the Working Data tab to see the data which will actually be used for any of the analyses you select within CAP. These data can be transformed or edited in a great variety of ways. Note that any alteration you make within the Working Data tab will not affect your raw data. Therefore, you can always revert to it if you decide that you've gone one step too far with your transformations. The handling zeros options are particularly useful for getting rid of samples that hold very few observations, or species or variables which are rarely recorded. Now I will show you how easy it is to allocate samples to a single group within CAP 5. This is useful so that they'll all be plotted with the same symbol or will be used together in a group test. Initially, all of the samples are unassigned. We will now select all the samples beginning with the letter A, because these are all from the River Avon, and put them into a group labelled River Avon. All the other samples will now be assigned to a chalk stream group. The Summary tab will give you summary information about the working data, the raw data, and even your groups. The summary tables are particularly useful when checking that your data has been loaded correctly. We have loaded a data set. Using the Working Data tab, we have square root transformed the data. Using the Grouping tab, we have made groups. And using the Summary tab, we have checked that our data looks right. Now let's go on and immediately do an ordination. With one click, we have undertaken a principal components analysis. Click on the PCA Plot tab and you will see a plot of your ordination. You can choose to plot eigenvectors, your scores, or both on the same plot. You can also decide whether or not you wish to plot labels. Note in this plot, all of the River Avon group are shown as green dots. Above the plot, there is a graphics toolbar which can be used to change almost every aspect of your plot. CAP also gives you the numerical results of any analysis undertaken. You can examine the distribution of individual species or other variables along each principal axis. The scree plot shows you how much the variability can be explained by the first few principal axes. Now we will undertake a detrended correspondence analysis. A number of methods within CAP will offer you a setup dialog box. You can, generally speaking, choose to use the defaults. As was the case for PCA, an ordination plot is available which can be edited in numerous ways. Click on the Grouping tab at any time if you would like to change the appearance of a group in a plot. We will change how the River Avon group will appear in any future plot. The River Avon samples now appear as large blue dots. We will now return to the plot to see the change. The final ordination method we will show you in this demonstration is multi-dimensional scaling. You can choose from a huge variety of similarity measures. As with the other ordination methods, an ordination plot is produced that can be modified in an almost infinite variety of ways. A particular feature of this plot is that you can show the magnitude of individual variables within each sample. First, select the variable. Then the level of transparency of the point. If you click on the variable list to activate it, then you can move up and down your variables very quickly using the arrow keys on your keyboard. This plot shows that the Acelidae are particularly abundant in the other chalk stream samples and not at all abundant in the River Avon. CAP offers 2 standard group membership tests. We will now open a second data set which holds botanical data and which we can use to demonstrate twin span. Note that we are using the Open a Demo Dataset drop-down menu to open these data. Twin span produces 2 dendrograms. One classifies the sites and the other classifies the species. This first dendrogram shows the samples. The indicator species are shown at the base of each split. This dendrogram shows the grouping of the species. The look of the dendrogram can be edited in a great many ways. CAP offers a variety of printing options for dendrograms. We will now open an archaeological data set to demonstrate discriminant analysis. Pottery shard samples were collected from 4 localities in Britain. Discriminant analysis demonstrates that the pottery from some sites differs substantially in its chemical composition. Now we can see where the Ashley Rails samples lie on the plot. Finally, to show the comparison features within CAP, we will return to the river invertebrates dataset. Use the Compare Sample option to compare the species found in different samples. Scatter plots can be used to examine the correlation between variables. The matrix plot is an excellent way to study the interrelationships between many variables. CAP has an excellent help system which will guide you through your analysis. This demonstration has only shown you some of the features offered by CAP. We hope it will get you started and that you'll enjoy using this program.